Good evening. It's my pleasure to welcome you to tonight's installment of Northwestern University's Center for Genetic Medicine Silverstein Lecture. This series is made possible by a generous gift by Herman and Bea Silverstein to the university to help promote the understanding of the importance and role of genetics in medicine. And tonight is a perfect example, I think, of what this series is trying to accomplish. We will be talking about personal genome sequencing, which is a new technology which is rapidly becoming a reality when it wasn't even conceivable 5 years ago. And it's likely to transform the healthcare system, so we need to understand it. And tonight we have a terrific expert to help us navigate the process. Our expert is Dr. Marc Williams, who's a board-certified clinical geneticist, and he is currently the director of the Genomic Medical Institute of the Geisinger Health System of Pennsylvania. Now, Dr. Williams also has a long history in helping work on policy issues and shape the policy of how genetics is going to affect the economics of healthcare. And he's served on numerous panels to investigate these issues, including panels sponsored by the Center for Disease Control, the Department of Health and Human Services, and also the American College of Medical Genetics. So it is my pleasure tonight to welcome Dr. Marc Williams, and he will tell us and help guide us into the understanding of what personal genome sequencing can and can't tell you about your future health.
Thank you. Well, thank you very much. It's a real pleasure to be here tonight on this balmy Chicago evening. I want to begin by giving a little bit of a perspective on the topic, and it's going to reflect my perspective. If you've been doing any reading at all, you're encountering a lot of different names that are being used to describe what it is we're going to talk about tonight. There's genomic medicine and personalized medicine and individualized medicine, and a term that you may not be as familiar with called precision medicine, and as Shakespeare might have said, what's in a name? And I wanted to spend a little bit of time talking about what I think are in these names and how that reflects how I think about this topic. So genomic medicine, and as was said, I direct the Genomic Medicine Institute, so in some sense this is the term that I am operating under in my job. When we talk about genomic medicine, it really encompasses a lot of different things. It encompasses what we traditionally think of in genetics, single gene disorders, so things like cystic fibrosis or sickle cell anemia or Marfan syndrome or things like this, the rare genetic disease, rare in the individual but relatively common in the aggregate. We're now moving into the area where we can actually analyze entire genomes. And when we talk about a genome, what we're really talking about is every single letter of the DNA strand that's in the cells in our body. So we're talking about literally about 6.2 billion letters. You may have seen 3 billion. 3 billion's on one strand, but DNA has two strands, so it's really 6.2 billion letters of DNA. And when we talk about the whole genome, that's what we're talking about. We can also analyze subsets of that. And we're going to talk a little bit more about this. I'm not going to define the terms right now, but we'll get to this, and I'll explain what I mean by other words like exomes and the pharmacogenome and this sort of thing. But I think it's really important right up front to remember that all of you have access to what is still the most powerful tool in genomic medicine, which is your family history. We can still predict more about your risk for disease based on what your family has experienced than any of the fancy techniques that I'm going to talk about, okay? So you've probably seen the advertisements that say, know your family history on Thanksgiving, you know, talk about family history. That's really still the best genetic test that we have in terms of understanding your risk of developing disease. So even though I'm not going to spend a tremendous amount of time talking about collection and analysis of family history, when I get to the end of the talk and kind of give you my vision of the future, I'm actually going to use family history as an example of how we can really transform how we practice medicine. Now, this is a definition from the definitive source of definitions, which is Wikipedia, of personalized medicine. Personalized medicine is the use of information and data from a patient's genotype or level of gene expression to stratify disease, select a medication, provide a therapy, or initiate a preventative measure that is particularly suited to that patient at the time of administration. Okay? Seems to be a reasonable definition. It's certainly a broadly available definition. But I would take issue with this definition, and it's a bit heretical for me as a geneticist to stand up here and talk about a definition of personalized medicine that doesn't actually explicitly talk about genetics or genomics. But I think it's really important that we understand that genomic medicine does not equal personalized medicine. And so the definition of personalized medicine I have chosen to use is one that was published by Steve Pauker and Jerome Cassirer in the New England Journal of Medicine in 1987, where they stated, personalized medicine is the practice of clinical decision-making such that the decisions made maximize the outcomes that the patient most cares about and minimizes those that the patient fears the most on the basis of as much knowledge about the individual status as available. Now, there's two things I really like about this definition. One, it's patient-centered. It puts the onus on us as physicians to understand what you, the patient, really care about. What are you trying to get out of this health intervention? What do you think is most important? And what do you not want to have happen? And that's not the same answer for every individual. And in general, in medicine, we have not done a good job of understanding what it is our patients really want and effectively communicating that and letting that inform treatment strategy. I think we're moving in a direction to do that better, but that's a critically important piece. The other thing that's important is this last bit, as much knowledge about the individual status is available. Okay? It doesn't say as much knowledge about the genetics or the genomics of the individual. It basically says any information that is relevant to the clinical condition is something that we have to pay attention to. And so genetics and genomics does not have a pride of place over any other information. And that's a critically important thing, particularly for us as geneticists to keep in mind. Now, the reality is clinicians have always practiced personalized medicine. In fact, one of my colleagues said, well, you're talking about this personalized medicine. Does that mean I've been practicing impersonal medicine? I said, in your case, yes. But in general, no. We have done personalized medicine. We try and individualize therapies. But what we're really talking about is currently intuitive medicine. We care for conditions that can be diagnosed only by their symptoms and only treated with therapies whose effectiveness or efficacy is uncertain. And then we wait for an empiric response, right? Take 2 aspirin and call me in the morning. That's, what we do. So you might call it empiric trial and error. Now, we try and be scientific about it, but the reality is that a lot of what we do in medicine is using this type of empiric treatment, watching for response, and we individualize the care based on how you actually respond. Now, bad for you if you happen to respond really badly to what we chose to do. We'd like to avoid those sorts of things. So what I'm really thinking about now is the opportunity to practice a type of medicine that Clay Christensen from the Harvard Business School refers to as precision medicine. And he defines this as the provision of care for diseases that can be precisely diagnosed, whose causes are understood, and which consequently can be treated with rules-based therapies that are predictively effective. And this is where I expect genomics to play a really key role, because a lot of this precision is going to take knowledge that is in the genomic realm. And so what we're going to talk about now really relates to how can we realize this vision of precision medicine, which has aspects of personalization, but I think is really where we want to be going. It's really not within the time frame available possible to talk about the new technologies in any level. It also presumes that I actually understand them at a level well enough that I could explain them to you. Fortunately, there are really smart people that know how to do this. But let me give it a shot. This is the traditional approach to genetic sequencing. So here's our DNA molecule, this double helix, where we have these A, C's, T's, and G's that pair up. I just have to say as an aside, it was in 1953 that Watson and Crick defined this structure, And the paper that they published was 1 page that described the structure of DNA. And there was one sentence in there that, to my view, is the most elegant sentence that has ever been stated in the medical literature. It says, "It has not escaped our notice that the proposed structure would allow for replication." I wish I could write like that. But unfortunately, it would take 2 pages to say that. So, at any rate, in traditional sequencing, what we do is we unzip these 2 strands, so we have a single strand. And then we use techniques where we use DNA that is primed with the different individual bases, and we can kind of reconstruct, using this as the template, we can reconstruct a second template, and if we then put this out onto a gel or some other type of visualization, we can actually say that this is an A, this is a T, this is a C, and if you've ever had genetic testing of any kind, this is the type of sequencing approach that would have been done to do this. So this actually does what we call genotyping, which is it says there is an A at this position in this particular DNA molecule. What we're now moving to is something called whole genome sequencing. What some people have referred to as shotgun sequencing, which tells you that mostly people with Y chromosomes were involved in coming up with this idea. So you take this big blob of genomic DNA and you blast it apart into little pieces that we can actually deal with, and then we amplify this and we create new strands, and then ultimately what we do is we genotype these individual strands, and then we try and align them so that we can create longer and longer and longer strands until we've ultimately recreated the entire genome, the 6.2 billion letters, 3.1 billion in one sequence. And so we have to assemble this sequence, and then we can generate that finished sequence. So when we talk about the Human Genome Project, it was not done with this technique for the most part. It was done using older technology. But now this is allowing us to do sequencing much, much faster at a much lower cost, as I'll demonstrate. So if we compare these 2 techniques, the traditional sequencing is slow. It's expensive, relatively. So if you do a genetic test, it's generally going to cost you between about $1,000 and $5,000 to do one gene. It absolutely determines the DNA base. It's highly accurate. It's clinically validated. And you can sequence virtually any part of the genome doing this, although it's very hard to sequence the entire genome very quickly. And this, as I said, is the method that we use for virtually all genetic testing. Now, if we look at whole genome sequencing, it's fast, it's inexpensive, and it's getting cheaper. We're literally approaching a cost of about $1,000 to do the entire human genome. To put that in perspective, the very first human genome that was generated from the Human Genome Project cost about $3 billion, okay? And that was less than 10 years ago. It does not actually determine the DNA base. It assigns a probability that at a given location there's a certain base there. So you can say, we have read this particular position 100 times, and we have called that base 98 of those times as an A. Therefore, there's a 98% probability that there is an A at that given position. Okay? That's really important to understand if you think about applying that into a clinical scenario. The accuracy is variable. There are parts of the genome where this works beautifully well. There are parts of the genome where it doesn't work well at all. And unfortunately, parts of the genome where we're really interested, like some of the genes that are involved in drug metabolism, this technique does not work well because there are a lot of genes that are very similar. There are what are called pseudogenes, which look like the real gene, but they don't actually function. There are duplications where you have extra copies, or you have deletions where you have missing copies. And this type of whole genome sequencing does not call that very accurately, and as a consequence, pieces of information that are critically important for clinical use cannot be generated using this type of a technique, at least today. And this technique is now just beginning to emerge into clinical use. So I think it's a legitimate question to say, with all those problems that I've just mentioned, why would anybody want to do it? Well, the real answer to any sort of a scientist is, well, why not? You know, we can do it, so let's go gaily forward and, push back the frontier. So I wanted to put this in perspective a little bit. I mentioned this earlier. So here's 1953, the DNA structure reported by Watson and Crick. And we kind of pooped along here. Here's where the polymerase chain reaction, which won a Nobel Prize, was reported. That was 30 years after the original structure was reported. In 1990, the Human Genome Project was initiated in the public sector, and then Craig Venter came along in the private sector and sort of threatened to say, you guys are going too slow, we're going to do it faster in the private sector. There was a meeting of the minds, and there was a public-private partnership that came together that really did allow completion somewhat sooner, although depending on who you talk to, the amount of credit or blame that is generated to Craig Venter versus Francis Collins is variable. The initial drafts of the human genome were published in 2001, and I should mention that one of the obligations, if you were a sequencing center, every night you had to upload all of the sequence that you had generated that day. So it was instantaneously available to anyone in the world that wanted to look at that sequence. So this was really public science. It was very exciting. And the first, the completed first draft, and completed is in quotation marks because it's really still not done, but the mostly completed first draft was reported in 2003 to mark the 50th anniversary of the DNA structure publication by Watson and Crick. It also happens to be DNA Day, April 25th, and it also is the 20th birthday of my daughter. So I was born to be a geneticist because my daughter was born on the day that they discovered DNA. So at any rate, it was in 2007 that we began to develop the real ability to do this massive parallel sequencing. So we're literally talking about now 5 to 6 years where this whole genome sequencing has exploded into practice. And the implication of this is, if we look at the cost per million bases, a megabase is 1 million bases of DNA sequence. Here's where we were in 2001, where it basically was about $7,000 to do 1 million bases. Here's where we are today, where we're basically under $1 to do 1 million bases. Now, the comparator here is something called Moore's Law, and if you're a computer scientist, Moore's Law has always been held up as the example of, sort of the optimal technology implementation, that, the cost of computer memory is decreasing at a logarithmic rate, and they held it up. So Moore's Law, we're kicking Moore's Law's butt, big time. So you computer scientists can't be nearly so smug as you used to be. But we're really literally now doing entire genomes. People are offering to do whole genomes commercially for $3,000 to $5,000. Which I think even the most optimistic of us thought was unattainable in this time frame. And frankly, we're now seeing successes emerge into clinical practice. Probably some of you are aware of the young fellow just north of here in Milwaukee who had a very complicated form of inflammatory bowel disease that was resistant to treatment, where they applied whole genome sequencing. They identified a mutation in an X-linked gene that caused an immunodeficiency. It gave them the first diagnosis of what was actually going on with this young fellow, and it informed the treatment that indicated that if they did a bone marrow transplantation to basically replace the immunodeficient cells with a gene that functioned normally, it would work, and he is now healthy today. As a result of understanding the underlying problem. And we've had several other examples of where this technology has been applied in a way that has been clinically useful. But these, for the most part, still are individual anecdotal experiences. So people have been saying, well, why should we do a whole genome? What's the justification? Well, we're now approaching the point where it's actually cheaper to sequence the entire genome than to do one gene. So if you wanted to have, if you had a family history of breast ovarian cancer and you wanted to have gene testing for BRCA1 and 2, that would cost you about $3,500 to $4,000 to get those 2 genes. You could have your whole genome done, including BRCA1 and 2, minus the patent issues, for the same amount of money. So if you can do the whole thing, well, then why not do it, okay? Having the information will save money. And I've heard people say, well, we'll do it through better prevention. avoidance of adverse events, and through more effective treatments, okay? So let's examine each of those reasons. And as Greg had said, I'm interested in healthcare economics and delivery. And there's a lot of the stuff that you hear about from the talking heads that really doesn't get at the true problems we're encountering in this country. So I'm going to try and do a little splaining about some of these myths that we hear. So the first question I was asked is, does prevention really save money? Well, there's an interesting study that was published in 2008 in Holland that studied the medical costs that were incurred after the age of 20 in a Dutch male. So these are in euros. If you're normal weight and a non-smoking Dutch male, you would incur $281,000 worth of healthcare expenditures for after the age of 20. If you're an overweight but non-smoking Dutch male, you would incur €250,000. If you're an overweight smoking Dutch male, you would incur $220,000. Anybody want to guess why? Yeah! You know, here's a dirty little secret. You know, you don't hear this. Dead people don't incur health care costs. Usually. Okay? So the idea that prevention saves money is really not true, in the sense that prevention, if it's done well, allows you to live longer, which means you can incur more healthcare costs. Now, if we look at it from the societal perspective, for society, it's a really good thing to have people live longer and to be productive longer. That's a benefit to society for any number of different reasons. But the healthcare sector does not recoup any of that benefit. It just expends money. It costs more. And so that's why we see this disparity, which I'll show you graphically in a bit here. Now, there are opportunities to avoid adverse events. And we talked earlier about pharmacogenomics, where we can use genomic information to predict response to a medication. So there are a couple of examples here where there are specific genetic changes that can predict the risk for an adverse event related to being on this medication. So if you have this HLA-B5701 and we give you a Abacavir, you're at a very high risk for developing a very severe skin reaction that can result in death, actually. So we really don't want that to happen. And if we can avoid these adverse events, then we're not only going to keep you healthy, but we're going to avoid a whole bunch of healthcare costs that are going to result from the fact that we gave you medicine that tried to kill you. And these changes do not have any effect on the efficacy of the medication. In other words, the medication works just as well, and there are alternative medications. The problem is that in most cases, the adverse events are related to the treatment effect. So if you think about chemotherapeutic agents, medicines that we use to treat cancer, the whole point of those medications is to kill cells, okay? What we're trying to do is to use medications in such a way that we kill more of the cancer cells than we kill of the normal cells. But for any of you who have ever been through chemotherapy, you know that we are killing cells. Your hair falls out because we kill off your hair follicles. You develop vomiting and diarrhea because we're killing off rapidly dividing cells in your GI tract. So these are predictable adverse events which we think are acceptable because we want to cure you of the cancer. Well, we now know that there are changes in your genome that can predict that you might be at higher risk for having an adverse event, okay? And so you would say, well, maybe if you have this change in this gene and we're using this medication, 6-mercaptopurine, maybe we would want to maybe not use that medication, or maybe we want to decrease the dose. But it turns out that the medication actually is more effective for the cancer that we're treating, because the cancer cells have the same genetic defect. They don't metabolize the drug well either, so it means it's more toxic. And so if we reduce the dose, what we're really doing is we're also reducing the effectiveness. So what we're really talking about is, where do we find the sweet spot between reducing the risk for adverse events and improving the efficacy of our treatments? And if we solely focus on adverse events, what we're going to do is to harm people by not giving them adequate treatment. It's interesting, there have been studies that have been done that show that medicine in general will kill 3 people by not doing something for every person that we kill by doing something, okay? So we'll kill 3 people by not giving sufficient amounts of that medication for every one, to avoid 1 person from dying from giving that medication. And there's a lot of different reasons why that could be, which we won't go into. But I think it's really important as we think about how to apply this technology to understand that many times it's a two-sided coin, that if we decrease adverse events, we're also decreasing efficacy. And if we don't remember that, we're going to cause more harm than good. So if we think about the overall economic impact of more effective therapy, there's at least the potential to decrease costs. I put a question mark after preventive measures because maybe prevention doesn't save money. Earlier interventions, well, if it's a very effective intervention and it's a curative intervention, then that could save money. If it's an earlier intervention but we're going to have to treat you for life, then it may not. We certainly want to have more effective interventions. In other words, we want to give you the medication right away that's going to be most effective for you, and we want to really avoid waste. We don't want to have to go through medication after medication after medication and try and find the one that's going to work for you. We'd like to pick that one first out of the box, and that will actually save money. But we also have the opportunities to increase costs because we're going to develop treatments for the untreatable. There are things that we're dealing with right now that are untreatable. And the little guy that I talked about where we did the bone marrow transplantation, tremendous outcome. He's cured. But a bone marrow transplantation is hundreds of thousands of dollars. So we've spent hundreds of thousands of dollars to cure one individual. We as a society have not as yet made decisions about what is the trade-off. How much are we willing to spend to effectively treat something? And in that case, we actually effected a cure. In many cases, we spend tremendous amounts of money to buy 3 months of life on average. And so where do we draw that line? And those are really societal decisions that we have not been able to really come to a good agreement on. If you live longer, which is a good thing for society, it consumes more medical costs. And pharmacy costs, particularly with some of the newer agents, are going up. And if there's one thing that we can all agree on in the United States is that we love drugs. I mean, if it's a choice between taking a drug or exercising, give me the pill. I don't want to go running and lose weight. If I can take the pill to lose weight, I want the pill. We also have to ask the question, is whole genome sequencing really as cheap as it seems? The costs that I showed you, which are incredible, really only counts the reagent costs, the chemicals that it takes to do it, and an amortization of the equipment. Okay? So that doesn't really reflect the true cost of using this information in medicine. Also, as you probably were able to determine without my saying it explicitly, the information that's coming off of these whole genome sequencing machines right now, in my opinion, is not of sufficient quality to be able to use in the clinic. We really need to validate using older style technology the things that we think are clinically actionable, because we're only calling probabilities that a change is there. We're not really measuring the actual change. And I think the clinical standard right now is to measure the change. It doesn't consider the cost of storage of the information. And I'm going to give you a little bit of a reality of healthcare economics and the information interpretation problem. So on those last three things, we're going to delve into some detail. So let me talk about healthcare economics first, because I think it's more fun. I like to think about this in terms of value. And value is not something that we typically talk about. We talk about cost and outcomes. We don't talk about value. And if we think about value, what it can really be crudely thought of as some sort of a relationship between outcomes and the cost of care. So it's a ratio of the medical outcomes achieved versus how much it costs us to achieve those outcomes. And if we think about outcomes from the perspective of the patient, well, there are medical outcomes. In other words, did we cure your disease? Did we prevent a disease? Did we avoid an adverse event? There are service outcomes, which is how many times did you have to take off work to go to the doctor's office to deal with this? How far did you have to drive? Did you have to come in on the Dan Ryan at 8 o'clock in the morning and sit there for 45 hours? What were the tolls? All of those sorts of things impact your service. And any of you that have ever come to a medical center, I guarantee you that the number one thing that if you have to fill out one of those surveys that you complain about is parking. Okay? Parking has nothing to do with medical care, but it's top on the service outcome list. Okay? So we have to pay attention to these things. How about the value of information? In genetics, because we've been relatively unable to provide treatments, we have said informing people about the problems that they have and the risks that they have and using that information to guide medical care is a really important thing. It's a very high value thing. And our patients generally back up and say, we're much happier now that we know what's going on, even though it hasn't really led to a direct treatment. But it's very difficult to value that economically. And so this has led to the concept of what's called personal utility. How much is it worth to me? And thus, how much should society pay? And it's been very challenging to actually get at this value. And the reality is that in medicine we do a very poor job of measuring the actual cost of providing a service. And if you've ever worked in a billing office, you'll know that what we charge for a service is highly variable, and what we get reimbursed for a service is highly variable. Presumably there's a cost of that service that is not variable, but we often can't determine what that cost really is. So I conceptualize that in this sort of a value plot, and this is very simplistic, so if you're an economist, please give me a pass. On one side we can put medical or service outcomes, and we can improve them, we can leave them unchanged, or we can make them worse. And we can also plot cost of care. We can decrease the cost of care, we can leave it unchanged, or we can increase it. Okay? And if we think about that concept, then we can think about green, yellow, and red. Okay? Here's the sweet spot. Improve medical outcomes with decreased cost of care. Okay? Probably the best example in medicine of this is vaccination. That we dramatically decrease the cost of care because we're preventing an illness that's going to consume a huge amount of healthcare dollars, and we're dramatically improving medical outcomes through that prevention. So highly cost effective. We can have situations and we can have discussions where we improve medical outcomes, but we actually increase the cost of care. So an example would be in chronic myelogenous leukemia. There was a drug that was developed that specifically targets a genetic change there. And it's the first effective treatment for chronic myelogenous leukemia that there has ever been. It's very expensive, okay? But almost everybody reimburses the cost. So as a society, we have said, yes, this increases the cost of care, but the improvement in medical outcomes is worth the investment in that cost of care. Now, there are going to be other situations where you say, well, we have a very minimal improvement in outcomes and a big increase in cost. Probably that's not worth it. Unfortunately, in the United States, here's where we spend most of our time, okay? We worsen the outcomes and we increase the costs, okay? Exactly where we don't want to be. But a lot of our new technology resides down in this area. And the best example of this was a number of years ago, some of you may recall that there was a lawsuit in California where an insurance company was sued for $80 million because they refused to pay for a bone marrow transplantation for a woman who had end-stage breast cancer. And they had determined that based on the evidence that this treatment was experimental and investigational. The court found in favor of the patient, actually the patient's family because she had died by that time. And overnight, every insurance company in this country covered bone marrow transplantation for advanced stage breast cancer. When the studies were finally done, not only did we find that it didn't work, but we killed people faster. Women were dying sooner that received bone marrow transplantation compared to those that did not receive it. So the insurance company, I hate to tell you this, was absolutely right. It was experimental. We did not have the evidence. It's estimated that between $500 million and $1 billion of healthcare expenditures were paid in this country to make women live less with breast cancer. Okay, and we do this over and over and over again. So that's, my friends, is why we're in some of the problems that we have. And if we want to demonstrate this graphically, here's per capita spending for healthcare. We're number one by a long way. $7,500 for the U.S. Norway is the next closest at $5,000. Japan is way down here. Those cheapskates, they're under $3,000. Okay, well you say, well great. We have more resources. We can spend more. Buying more healthcare is a good thing. Well, if we think about it from value, though, we look here. And what are we getting for our money? Well, we're number one in healthcare expenditures. Life expectancy, we're number 17. Infant mortality, we're number 17. Potential years of life lost, we're number 17. Japan, our cheapskate friends, are number 1 in life expectancy, number 2 in infant mortality, and number 2 in potential years of life lost. Now, if you think about what you're buying, we're getting terrible value for the money that we're spending in this country. And that's the dirty, dark secret that no one is bringing up in this healthcare debate, because we're much more interested in talking about the ideology as opposed to the reality, which is we're doing a very poor job of delivering high quality care at a reasonable cost. Now, if we apply this to genetics, it gets even more embarrassing as a geneticist because we have almost no information. Now, some of this is because we have the challenge of the rare disease, which is how do you get enough of any rare disease to be able to understand what would be an effective treatment? But you know, while we can perhaps excuse ourselves, the reality is we have not done any sort of concerted effort to gather information in a systematic way to be able to answer those questions. And so as I stand up in front of my colleagues and I say, you know, we are a faith-based specialty, ladies and gentlemen, we go in and say, you got to believe that what we do is good. You know, in God we trust. In genetics we trust. All others should bring data. We should bring data. We should be generating data. We've been treating phenylketonuria and inborn error of metabolism in this country for 60 years through newborn screening. We identify it in newborn screening. If I stood in front of this many metabolic geneticists and I said, what is the agreed upon level of phenylalanine, the target of the treatment? What is the optimal level of phenylalanine? What is our treatment target? We don't have it. We would have 18 different answers. We would have as many different answers as we had metabolic geneticists in the room. After 60 years, we should know the answer to that question, because I can tell you what the hemoglobin A1C should be treated to if you're diabetic. I can tell you what our blood pressure target should be. We've done a poor job. And frankly, our system is moving to value-based assessment, so we have to figure this out. And ultimately, that means we have to figure out the value of whole genome sequencing. So as I think about this, I think one of the opportunities that we have is how could we reuse genomic information over the course of a lifetime? Because if you spent that $3,000 to do a whole genome, but you could use that information, since it doesn't change, if you could use it over that individual's entire lifespan, now maybe we could deliver some value. So I just wanted to show a little bit of, from different perspectives in the lifespan about how we might do that. So we could conceivably do it in the prenatal area. So we do prenatal screening now for certain genetic diseases like cystic fibrosis or diseases that are more common in the Ashkenazi Jewish population. We could do whole genome sequencing and basically look for what are the diseases that could be present in a fetus that could cause potential problems. Maybe there's a known genetic disease in the family that we want to test for. We could also use it for diagnostic purposes. If that fetus is showing us problems, if it's not growing normally, if there's a congenital malformation that we can see on ultrasound, then we could use this. So if we obtain the information, then we could store it and potentially reuse it later. We could use it in the context of newborn screening. Right now, we get these blood spots and we do some analysis on them. Some of it is genetic analysis. A lot of it is measuring analyte levels, chemical levels in the blood. But we use genomic approaches for disorders that we screen for, like deafness and for immunodeficiency. And when we're talking about deafness, we're talking about 150 to 200 genes. Immunodeficiency, probably a similar number of genes. So this would be a natural place where you could say it would be cost efficient to obtain a whole genome sequencing. Just look at those deafness genes for a child that failed hearing screening, or just look at those immunodeficiency genes for a child that failed immunodeficiency screening. We'll save the rest of the information and use it when we need it. And of course, we might use it diagnostically for a neonate that has malformation or some other form of severe illness. Well, we could use it in children. Children will be given medications. In some cases, those medications, the metabolism of them can be affected by genes. We could use that information to better inform which drugs we should be using and at what dose. If they had to go in for surgery, we could look at genes that predispose to reactions to anesthetic to try and avoid those. And we could use it in diagnostic contexts, autism and developmental delay, malformations, neurologic diseases like seizures and the like. Young adults, adolescents, again, medications. And now we're beginning to talk about the risk for adult onset disorder, where we can talk about individualized prevention. Do you have a genetic mutation that predisposes you to breast ovarian cancer, where we might want to initiate mammography at an earlier age? Do you have an increased risk for colorectal cancer based on genetic features that would say we would want to initiate colonoscopy at an earlier age? Things of that nature. We also know that young adults tend to make littler children. And so in a preconceptional setting, somebody contemplating a pregnancy, could we do genetic testing on the mother and the father to say, here are things that you might potentially be at risk for that we would want to look out for? And we certainly would want to use this in the diagnostic realm for rheumatologic disease, cancer, early onset adult disorders. And then as we age and are exposed to more and more medications, this genomics that relates to drug metabolism becomes more and more important. Again, we want to talk about that individualized prevention. There are diagnostic opportunities. But also, as more common diseases develop, like diabetes and stroke and cardiovascular disease, there may be subtypes of those that can be defined genomically that would lead to different treatment approaches depending on which subtype you fell into. We might be able to use this for prognosis to say, if you were to develop diabetes, what's the likelihood that you're going to have progression of complications of diabetes like vascular disease or eye disease? And then ultimately, we would use the germline genome to be able to interpret genomes that have undergone catastrophic changes leading to cancer. So I think you can see there would be plenty of these clinical opportunities to use this information that we would only have to obtain once. And I think if we can figure out how to do that, then we would really be able to extract the maximum value out of obtaining a genome. Now, again, the advantages of doing this over the lifespan is we would spread the cost out over the lifetime of care. We would not have to repeat testing over and over and over again. We would have the information available so we could use it as soon as it was needed, as opposed to ordering a test and waiting for the test to come back. We would allow more precise pharmacologic therapy, hopefully to avoid adverse events and choose the best tolerated, most effective therapy. But there are a lot of questions. And I'm not going to read these because we're going to just kind of address each of these very quickly. Storage of information. If we were to print out the genome onto pages of paper, initially it was estimated it would be the height of the Washington Monument. It turns out that was a little bit of a pessimistic estimate. It turns out we only need 172 copies of the New York City phone book to print it out in paper. So you can imagine your doctor walking in with that medical record, right? So the whole genome, as I said, 6 billion base pairs, and so the cost of storage is significant. And in fact, with the way that sequencing costs are dropping off, some people have been making the argument that it may be cheaper to store the information in the DNA molecule and just go back and sequence the molecule when we need it, as opposed to storing it somewhere in a computer or in the cloud. That still remains to be a debate. But I think a more pertinent question is really, do we need to store all 6 billion bases? And so I mentioned this term exome before, and I wanted to come back to it because this is a subset of the genome that is potentially more usable. So when we talk about the exome, what we're really talking about is DNA that makes up the genes, and the genes themselves are the ones that actually produce proteins, the proteins that produce structure and function of our body. And that's the things that we tend right now to understand the most. And then there are also regulatory regions for those genes that are included in that. That's only about 1.5% of the whole kit and caboodle. So that breaks it down to in the range of about 90 million bases, so that's much more manageable, of course. But mostly that's where the clinical relevance is. The reality is most of the exome still isn't usable. We don't understand what most of it means, so there's probably even a smaller subset that's usable. And the current approach is, if we're just looking to sequence just the exome, actually misses about 10 to 15% of the exome. And if you miss the important bit, that's not such a good outcome. And ultimately, as we learn more and more and more, there's going to be parts of the genome that are not included in genes that are going to be critically important. And so in some sense, we don't want to lose that information if it's going to ultimately become important. So the concept that we've been talking about at our place is what we call the clinical exome, where we would store only the information that currently has evidence of clinical effectiveness. It's a small subset of the exome, All of the information would be of potential clinical relevance. And as we accumulate more information, as we learn more, then we can add additional bits into this clinical exome. And in fact, we can add it from any part of the genome that proves to be clinically relevant. Now, to give you an idea of what we're talking about here, this is from a paper from a colleague of mine. And when we talk about, he talks about how we might be able to bin different genes. So this is focusing on genes specifically. But right at the present time, we have a handful of genes, probably under 100 genes, that not only do we know what the genes do, we know what diseases they're associated with, but we actually have something that we could potentially do if there's a change in the genes. So some of these are the cancer predisposition genes, other things of this nature. And so we have a very, very tiny amount that where if we saw a problem in that BRCA gene, we would know exactly what to do. So right at the present time, that would be a very small subset that we could manage, and we could actually manage it reasonably well. Now there are these bin 2s. 2A is low-risk information that we might get incidentally, so some of the genes that are responsible for drug metabolism, for example, where we're probably going to be getting into the maybe hundreds and low thousands. There are going to be some of them where we find things that have a very high risk for something that we can't do anything about. And so this would be something where we would want to have a conversation to say, we found something, it's pretty important, but we can't do anything about it. Is that something you want to know about or not? Okay, or maybe we don't ask, I don't know. We're still working on that one. The bottom line is bin 3 is, that's where most of the genome goes right now. We don't know what the hell it does. So we don't want to pay attention to it. We don't want to make medical decisions on something that we don't understand, because that's going to incur cost. It's not going to improve outcomes. And so what we're currently working on now from the clinical side is how do we define that critical minimum set, that clinical exome, that's actually usable information. Now the next problem is availability of information. This information has to travel with you. It has to be available whenever we need it. Now I know all of you in the healthcare system never have any problems with your records getting to where your appointment is, right? That doesn't happen here in Chicago? Okay, I'm hearing laughter, so I'm assuming there may be some issues there. We don't do a good job of moving information even around in one healthcare system, much less between healthcare systems. So right now, if I got sick and I got treated here at the hospital, there would be no access to any of my medical records at Geisinger or at Intermountain or at Gunderson and all the other places that I've been treated, because they all reside in those institutions. They don't follow me, they just sit in the institution, and they're of little use to me if they're sitting there. So we need to come up with some solutions that allow us to move information. And this is not specific to genetics, all right? This is any kind of healthcare information. So whether this results in sort of a national electronic health record where we can exchange information, whether there's a centralized repository of genomics, if that's the case, who would be the trusted broker? The government? Not in Idaho. And then what would be the security that would surround that? And who would have access to it? And under what circumstances would there be access? Or is this going to be a personal responsibility? Are you going to have a little flash drive? Is it going to be something on your phone? Are you going to have an implant where they just kind of plug you in? You know, have a USB port, you know, and you're, you know, draw blood and power up your computer from the one port? I don't know. But we have to figure this out. Evidence is a big problem, and I kind of went over this before. So we have very, very few examples right now where we have information that we know can affect clinical care. Now we anticipate that there's going to be a rapid increase in knowledge, but we have to be able to aggregate the data. So those of us that are doing research, if we learn something, everybody has to know about it right away. And so we're working on solutions where we can share data very rapidly, so we can rapidly improve our information. But at the present time, we don't have any sort of robust, centralized way to accumulate and store the information, annotate the information, and more importantly, make it available to electronic health records so that they could consume that information. One of the things that Geisinger is working with Northwestern is on this eMERGE network, which is the electronic medical records and genomics. And these are the types of problems that these organizations are beginning to work on to try and say, can we come up with solutions to deal with these problems? Now, genetic discrimination, frankly, has been of great intellectual concern, but the reality is there have been very, very few examples of true discrimination, either in health insurance or in employment, based on genetic information. That being said, it is still one of the #1 concerns of patients and physicians whenever we talk about using genetic or genomic information. I would argue that we've gotten everybody so head up about this issue that we're actually harming people more because they're scared to use the information, and they're having harm because we're not using effective information that we could use to improve their care. There is national legislation, and in most states, legislation that protects you from having your genetic information used against you either for health insurance or for employment insurance or for employment. Life insurance, disability insurance, not covered. But for health insurance, for employment, there are national coverages, but still a significant concern. So ultimately, this is my graphical view of the strategy, that we have all of this knowledge that's coming out of the basic sciences, and we have to develop an intelligent filter so we can take this vast amount of information and get it down to the small amount of information that we can use that's ready for prime time. And then we have to be able to take that into some sort of a black box that is going to involve informatics and computers, electronic health records and the like, to be able to make sure that that information is effectively used, and ultimately we want this to be used to hit the 6 quality outcomes that we've agreed on. Avoid adverse events, improve physiological function, relieve signs and symptoms, improve well-being and functioning, achieve patient satisfaction. Actually, we could do that if we could do the parking thing. And minimize the cost of care. Okay? So ultimately, this is what we have to think about for anything that we're doing. And if what we're doing doesn't affect one or more of these, we should stop doing it. The good news for me is that we haven't figured out how to do any of this yet, at least not robustly. So that's job security as far as I'm concerned. So to realize precision medicine, we've got a lot of challenges. Okay? We've just gone through them. So how can we really individualize this at the point of care? I mean, what's the reality here? Well, my view is that, and this is not just mine, I should say, but that we can use techniques of mass customization and say, okay, well, what's that? Well, here's one example. What color phone do you want? Okay? Or you can design your own t-shirt. Okay? Go online. Or this one I thought was really cool because they got these slider bars. I mean, there's an infinity of colors that you can pick to design your own hoodie and cut the arms off and you could be Bill Belichick and coach the Patriots. But probably the best example that we deal with in our daily lives is our personal computer. Okay? We get exactly the computer that we want. Okay? And it's not some little old guy in the back room building it. It's built on a mass assembly line, but using these mass customization techniques. So we get exactly the computer that we want, but a high reliability. And so this is really what we've gone from. In the 19th century, we had the craft. Very high quality, customized, but high cost. If you watch Antiques Roadshow, they say, this is a Philadelphia chest. Okay? It's a beautiful chest. We can tell it's Philadelphia because of this, this, and this. This is a Delaware chest. Okay? Very high quality. We can tell it's a Delaware chest because you couldn't, you know, it was 30 miles from where you lived. That was the kind of chest you got. In the 20th century, Henry Ford came along, mass production, very low cost, wide distribution, but variable quality, generally probably lower quality. But in the 21st century, mass customization allows us to have customized goods at very high quality because we're using reproducible techniques, very low cost, and wide distribution. So, you know, the iPad that is assembled and built in a factory in China is built to exactly the specifications that you want, so you can stand 3 hours in line to get one. That's the choke point now, is actually standing in line to get one. Now, there's a lot of problems with applying this to medicine. Our evidence for therapies are almost exclusively population-based. We take a whole bunch of people and we give them a medicine, we say, do you get better or not? And the problem is, and the FDA requires us to do that, by the way. So the problem is you say, well, you know, if a certain number of people get better, then we'll approve it. If they don't, then we don't approve it. But the problem is that in a lot of things, we have a small number of people that get really better, and everybody else doesn't respond. So how do we figure out those 3 people out of that 100 that really respond to this medication and make sure that they get it? But how do we design studies to do that? Currently, our electronic health record systems do one thing really well, and that's generate the bill that you get. But they do not support aggregation of relevant patient data at the point of care, for the most part. we have a problem with our brain, which is the brightest human being can consider 5 variables at any given time. I just told you that there are 6.2 billion letters in the genome, okay? So what's between the ears here is not going to scale, particularly if we start to add in all the other information. So we have to figure out how to manage the data and compress it into such a way that it's reliable, but it can actually be acted upon by a human being. We are very poor at collecting outcomes data from the real world. So we do a clinical trial in highly controlled environments. We see that something works. We put it out in the real world, and it doesn't work at all. Why? Because, you know, people don't take the medication, or the doctor doesn't use it at the right dose, or all these other things. And so how do we really know how something works in the real world as opposed to a highly controlled clinical trial? And then our traditional methods of disseminating knowledge, like journals and actually talks like this, are highly ineffective for dealing with the scale of information. So sorry to inform you this late in the talk that it was a waste of time, but there you have it. So how might we do this? And so this is sort of a little thought experiment that used some tools that we had built where I used to work. And I just want to demonstrate what I think might be a healthcare encounter in the future, and I promised you I was going to do it using family history. So what we're looking at now is a patient portal. So some of you that are getting your healthcare in systems that have electronic health records, you may have a portal where you can communicate with your doctor, schedule appointments, or whatever. So this is a patient view, and there's a family health history that's available to be filled out by the patient. And so Grant, my patient, has gone in and has filled out his family health history. And he has the opportunity to print it out, and he also has the opportunity to click a button that will send it to me. And so he clicks that button, and when I open up my electronic health record, so now I'm in my office and I open up my electronic health record for the day, and I have this list of messages, things I have to deal with. And I see I have a message from my patient Grant, and it's a family health history message, and it says this patient has logged into their patient portal, they've completed a family health history, and it shows an increased risk for a familial disease. It's available for your review. You can view it as a pedigree, like we saw previously, or you can view it as a table. And we turn out that most doctors like to see the information in a table. And when we see this, what we see is, oh, father and brother had heart disease, and there's also stroke in the paternal grandfather, and there's prostate cancer in the maternal grandfather. And I say, okay, all right, well, what should I do with that? Well, I could upload this to the family history web form so that the data would become structured data, or it says I can perform a risk assessment. So if I click that button, I get this big box. It says, increased risk for coronary artery disease. And why? Because there are 2 first-degree relatives, the father and the brother, that had premature coronary artery disease, which is defined as coronary artery disease under the age of 55 years. So I can add this to the problem list. So if I click that button in the problem list, it would say family history risk for coronary artery disease. So it would remind me that every time I see Grant in the office, I would look at that problem list and I should remember to talk to him about that. But it also says you can assess this cardiovascular risk using clinical data. So if I click that button, it accesses data from previous clinical encounters that Grant has had in the system, including his blood pressure measurements, his cholesterol measurements, and other things like does he smoke cigarettes. And here it flags some abnormal findings, that he has high blood pressure and that he has a low HDL cholesterol, and it gives me the definitions of this. And what it says is if we incorporate this clinical information with the family history, Grant has a moderate cardiovascular risk because he has more than 2 risk factors for cardiovascular disease. So you should consider doing an inflammatory marker test to reclassify the risk. And if I click that little button, it shows me that if this inflammatory marker test, and they're defined here, is not elevated, that I would want to treat Grant with medications to get his LDL to under 130 milligrams per deciliter. But if it's elevated, that means that he's actually at high cardiovascular risk, and we should be much more aggressive with treatment. Now, I could schedule an appointment to have the patient come in, but I say, geez, he just was in July. I probably don't need to pull him out of work and have him drive down. I can order the test and notify the patient. So I click that button, and now we're back on the patient portal. And now Grant has a message, and he opens that up, and it's from me, and it's about the family health history. We've reviewed your family health history. We identified increased risk for cardiovascular disease in your family. This information, when combined with your personal risk factors, places you in the moderate risk category for cardiovascular disease. There's a blood test that can help us determine treatment goals that can reduce your chances of having a problem. I've placed an order for this test in the system. Please contact our scheduling clerk to make arrangements to have the test performed. I will contact you with the results. Call or email if you have questions. So here we have a very sophisticated medical encounter that never involved the patient and the physician actually sitting in the same room and talking to each other. And this is based on evidence of best practice, and it's something where now the testing could be done at the patient's convenience, and we could work out a treatment plan at a much more efficient way, as opposed to coming in and saying, well, how are you doing today? Do you have any concerns? Well, I don't know. And I ask a couple of questions about family history, and maybe I'm smart enough to recognize that there's a problem, but I don't remember the evidence and all this sort of stuff. So I think ultimately this is how we're going to be dealing with this information. And you could see how this type of an approach could also pull in genetic and genomic information. So I would contend that we're on the verge, really, of a revolution in medical care, and I don't think that's an overstatement. Genomics is going to play a part of it. It's not because of genomics, but it's because of the things that we're going to need to do to manage genomic as well as other information that's going to really affect this revolution. But we've got a lot of challenges that are going to slow the pace of change. I have to say this because I always do. So how many physicians does it take to change a light bulb? Change! Okay. So ultimately, this success will be measured by enhancing the value of healthcare provided as measured from the perspective of the patient. And I think, again, I want to return to that. I started with it, I want to end with it. Ultimately, if we don't listen to what you guys are telling us about what you care about, all of this fancy stuff is going to fail because we're never going to be able to match up what you want and what we think is going to be best. So with that, I will end. I know I've gone just a tad over, but I think there may be some time for some questions.
Thank you, Dr. Williams. Before we go to questions, I just have a few small notes that we have to go through. So there is food and beverages outside. Dr. Williams will be staying to, well, he probably won't get a chance to eat or drink anything, but you guys will, and you can pepper him with questions. We will do questions for a few minutes and then move outside. Before we do, I'd like to just thank some people who are involved in tonight's presentation. First, again, Herman and Bea Silverstein, who donated the money to make this lecture series possible. I have to thank Dr. Peter Kopp, who's director of the Center for Genetic Medicine, of which I'm a part, and which puts on this series, and the staff of the Center for Genetic Medicine, in particular Michelle Mahoney, who worked very hard to get this organized and make it all come off correctly. Thank you.
Perfectly adjusted for you, I can see already.
Yes. Doctor, I'm a nurse and a nurse educator, and I have a question and a comment. My question relates to what area of our society is really taking the gauntlet for human genetic and genome research? Is it the government? Is it pharmacy? Is it industry? And in your mind, what's the, who should be the major stakeholder?
So the answer to your question is yes. Everyone's involved. And the answer is that everyone should be involved, because I think if we've learned anything, it's that the broad perspective of a variety of stakeholders, and I would include in that also patient-generated research, is critically important if we're going to move forward. If we leave this to just one sector, then we're going to get a slice of the whole pie, but we won't get the whole pie. So I think this has been, the Genome Project, I think, because of its public-private nature and its public availability, has been an excellent model that has been something that has been held up so that at least the government-funded research, there's an obligation to deposit data so that everyone can really generate it. So it's really going to be something that I think a lot of crowdsourcing is going to be involved with, and ultimately I think that will speed progress.
My comment is this. As a nurse and someone who used to be involved in utilization research, I know that right now in the U.S. we spend the large share of the Medicare dollar an end-of-life care to people who are terminal, and yet we continue to try to treat them.
Right.
That is such a far range from genetic testing of large populations to do prevention and intervention when we can. How are we going to make that shift?
Yeah, so I blame Dylan Thomas. Rage, rage against the dying of the light, right? You know, we just want to hold on with our fingernails. But, you know, really this comes back to where I ended, which is I think in many, many, many of those situations, we've never had the conversation with the patient about what do they want and what would they want to avoid. And I think if you ask most people, and actually the researchers that have asked people about end of life, say, do you want to die in the hospital connected up to a bunch of tubes and machines, or would you like to die at home with your family around you? You know, everybody wants that second, but if you don't communicate that well, then we can't do that sort of patient-centered approach. And so this is not an either-or, but it reflects the broader problem that we have in society about where is it most appropriate to expend resources, and how do we have the conversations about the appropriate use of resources, and whether those be at an individual level or whether those be at a societal level. And of course you remember what happened when we tried to have a national discussion about that very topic, right? Death panels. We're gonna, you know, we're gonna kill you because you're gonna cost you money. So, you know, so we have all these ideologies that we have to deal with, and things that people bring. But the point that you bring up is extremely important. That if we look to technology to solve the healthcare problems, we will not solve the healthcare problems, because the vast majority of healthcare problems are not technological. Thank you.
Thank you.
Hi, I'm Barbara Marriott, and I'm a former correspondent with Time Magazine that worked on healthcare matters. I'm very interested in the genetic sequencing of and diagnosis and treatment of Steve Jobs. I was wondering whether you were familiar with that.
Not personally. I mean, I do know, and it's not only Stephen Jobs that had this, but there were a number of other individuals who have advanced or end-stage or otherwise untreatable cancers that have approached the National Human Genome Research Institute and others to say, I would like to take advantage of this new technology to see if you might find something that would allow a novel treatment to be tried. So there have been a number of examples where this has been applied, and there was actually a 3-part series in the New York Times, I think within the last month, that in the area of cancer described 3 patients who basically went through this technology in the context of an untreatable cancer, and kind of presented 3 different perspectives. And I would definitely refer you to that. But this is, you know, what we're doing right now is we're applying this in sort of these individual situations where we sometimes learn interesting things, but it's not at a level where we're aggregating our knowledge very effectively. And that's what we really need.
Well, my understanding is that they sequenced the genes of the cancer
Correct.
itself. What does that mean?
So the genetic material of the cancer, DNA, just like the genetic material in the rest of our cells, in cancer there have been damage to the DNA that has disabled important parts of the DNA, the genes and other types of regulatory elements that basically allow the cancer cell to grow wild. So it genetically looks very different from the rest of the normal cells. And so if you do sequence the normal cells and sequence the cancer cell, you can identify the differences in the cancer cell compared to the normal. And some of those differences occur in genes that we know are responsible for growth, and some of them occur in genes where we actually have drugs that could potentially target. And so that's the point, is that if we can identify targets from that sequencing of the cancer, that may make us smarter. And I would say that if we're looking in the near term for where we're really going to see applications of this broadly, it's going to be in oncology, because I think we're really reaching a period where we're moving away from trying to say, is this a breast cancer or a colon cancer or a skin cancer, to saying, what are the molecular reasons that this is a cancer, and what are the treatments that we can apply based on the molecular definition of the cancer as opposed to where it arose. So we'll have to see how that plays out, but at least that's my prediction, and I guarantee my predictions to be 100% wrong or your money back. So, as Niels Bohr once said, prediction is difficult, particularly when it involves the future. Now, most people attribute that to Woody Allen. I mean to Yogi Berra. But it was, in fact, Niels Bohr, and it just goes to show you that when Yogi Berra said, "I've never said half the things I said," that he was right. So,
Dr. Williams, I came out on this balmy evening because you impressed me at the Institute of Medicine at the roundtable on genomics and how we were going to economically implement it.
yeah, I apologize for that.
Well, so many strands I want to pick up on, but for the sake of this audience, what I'm curious about is when you look at the architecture of information, and you have this down cold from Intermountain, but then you alluded to the fact that so much of what we do in medicine is not only beneficial because of empathy and intuition, and what I draw upon there is the studies that were done with anthrax. Everyone remembers when we had the U.S. postal scare. And there were two physicians who saw two patients. And one decided the patient was well enough based upon the symptoms to go home. And you're nodding your head, and you remember that the other patient, the physician had the good intuition to hospitalize that patient. And that patient was treated with Cipro and survived. So no matter whether or not we look at the economics of genomics, we have a failed healthcare system. And another example, and when I'm done with these two examples, I want you to say, how are we going to get to that point where we have that dialogue digitally and the patient comes in? So if I may, the second example is when we look at something as simple as getting a patient to understand health outcomes. That if we do this, the more likely possibility is you're going to feel better. Yet our healthcare system is set up that right now we have to prescribe a drug that is cheapest, even if we know there's a better drug that's going to get that patient that health outcome that's so meaningful for them. So we have opposing levers, and when we have this ability, we're going the wrong direction even more. Right. So how do we rationalize this, and what is Japan doing that we're not doing independent of the technology?
So it's really more what Japan isn't doing that we are doing that relates to the cost. But I mean, it's basically elimination of waste and reducing access to technologies that are of marginal benefit. And some people refer to that as rationing. I think it's rational rationing. But so how much are you willing to pay to get an incremental benefit? And at some point you get out to the law of diminishing returns, where we spend more and more money and get less and less and less. And that's where we're sort of in that tail in the United States, where most other countries are in the area where they're really expending their dollars on things that have the biggest bang for the buck. Now, the points that you bring up are just 2 points of what are almost an uncountable number of unreconciled incentives in our healthcare system. We do not have alignment of incentives. So as physicians, we're paid to do more. We're not paid to do less. And so if we do more, we get more money, which means we're inclined to do more. So if we're on the borderline, should we or should we not do it, we'll do more because we actually make a bit more money that way. But from a societal perspective, that's the wrong way to go about doing things. We have to deal with all of these different issues of the poorly aligned incentives. And one of the reasons that I practice in an integrated healthcare delivery system where we have providers, where we have hospitals, and where we have an insurance plan is that we can set all of the people around the table and have intelligent discussions about these types of issues. So that if we have a question to say, well, geez, our formulary says we use this drug because we get the best deal on it. But I can show evidence that says, for this group of patients, 10%, you're much better off using this drug. And for this group of patients, 5%, that drug is going to make them sick. They will say, you know what, let's get that information available to the clinicians and we will adjust our formulary so that we pay for the right medication. I would predict that this, as we build it out, will mean the death of formularies in this country, because you cannot justify using a population approach if you have better information that you can use individual information. So that's, I mean, this is a topic, as you well know, that has been debated for years, so we're not going to solve it tonight, but that's a few brief thoughts.
Thank you. Oh, okay. Hi. So this is kind of in relation to the first question that was asked in regards to where the research lies. This might be my lack of knowledge, but you briefly mentioned patents in regard to the BRCA1 gene, and are there going to be certain more cost effective? It's going to be more expensive because certain private industries are patenting certain genes that they find are correlated to certain diseases, and like the effects of that on the costs.
Right. So you can think about this as, you know, basically parsing out and patenting, you know, the genome. And so that you can imagine the challenge as a clinician that you would have is if you wanted to get information from the genome and you said, well, geez, you know, Myriad owns this part and Athena owns this part and University of Chicago owns this part and Northwestern owns, you know, you wouldn't be able to navigate that. And in some ways it would be like buying I almost said a VCR. I almost showed my real, okay, I literally came this close to saying that. Actually, I did say it technically, but it would be like buying a Blu-ray DVD and saying, okay, well, to buy this, the laser is patented by Panasonic. The interpreter, the vision, that's Sony. And you have to buy this part from this company and this part from this company and this part. And so the way that the electronics and other industries have dealt with this is to deal with a patent pool, where all of the people that contribute to a product get together and say, we've all got a stake in this, we're going to pool our patents to allow the product to come to market, and then we all get a little piece. Now that approach has been tried in genetics and genomics unsuccessfully. But I think there's also been a move away, and one of the big benefits of the whole public knowledge stuff is that as things get blown out into the public domain, then you can't patent it. Now I think there are things that should be able to be patented. If you develop a unique test or therapy, you know, and you're deriving something from the genomic information that's novel, then that's something you should be able to have some remuneration for. I mean, you've put in the effort, you should get paid for that. But to just say, here's this sequence of DNA and we're patenting it, and if you want to use it, I mean, it's like, you know, I own Park Place. And if you land on it, you're going to have to pay me $150. I think Park Place is $150, isn't it, without hotels? At any rate, you know, that just isn't going to sustain. And so the good news is that I think that there's not nearly as much enthusiasm for doing that. Most of the information is out there, so there's really no mechanism to do it. It'll be interesting to see what the Supreme Court says now that that Myriad case has come back to them. The case has been, there have been related cases where the court has actually been relatively averse to the idea that you can take biological measurements and patent them, basically saying this is a product of nature and a product of nature cannot be patented. So it'll be interesting to see how that sorts out. But I think in the long run, this is not going to be a huge impediment.
So I know there are a vast number of questions that people would like to ask, and many of these verge into areas of policy that we could spend all week discussing. So let's just thank Dr. Williams again, and anyone with questions can talk with him during the snacks.